Here is my central dogma of biology. So I wanted to start out with a sort of general idea of what the central dogma is. So the central dogma of biology describes a two-step process of transcription and translation by which the information in genes flow into proteins. So it goes from DNA to RNA to proteins. Replication, transcription, and translation are the three main processes used by all cells to maintain their genetic information and to convert the genetic information encoded in DNA into gene products, which are either RNAs or proteins. So the three main processes are replication, transcription, and translation. So what I have here in the picture, just a general overview, we'll go deeper into it later, is your DNA, which is transcribed into mRNA, which then leaves the nucleus to be translated into polypeptides with the use of ribosomes. So the first step is transcription, and this happens in the nucleus. So transcription is when one strand of DNA that makes up a gene, called the non-coding gene, or the template for the acts as a template for the synthesis of a matching RNA strand by an enzyme called RNA polymerase. This RNA strand is the primary transcript. The primary transcript carries the same sequence information as the non-transcribed strand of DNA, sometimes called the coding strand. However, the primary transcript and the coding strand of DNA are not identical, thanks to some biochemical differences between DNA and RNA. So the important difference is that the RNA molecules do not include the base thymine, instead it has uracil. So it goes from the 5' to the 3' direction. The enzyme that catalyzes this is RNA polymerase. So the strand that does the coding is this one, coding strand. And this is the non-coding strand, or the template. So we'll just call it the template strand. And this is the RNA that is getting transcribed. So your result is your primary transcript of RNA. So as you can see, this would be the same as the coding strand, but the only difference is that thymine is replaced with uracil. So I also wanted to add the fact that there are 4 types of RNA that are each encoded by its own type of gene within the DNA. So here's your DNA strand, and you can have tRNA gene, protein coding gene, rRNA gene, and an snRNA gene. So these genes within the DNA code for specific RNAs. So your tRNA gene will make tRNA. Your protein coding gene will make mRNA, which we'll use in translation. And then your rRNA gene will make rRNA. And your snRNA will make snRNA. So the purpose of your tRNA is to is a transfer RNA, which brings the amino acids to ribosomes during translation. The purpose of your mRNA is to encode amino acid sequences of a polypeptide. The purpose of your rRNA is to make up ribosomes. And the purpose of your snRNA, which stands for small nuclear RNA, forms complexes that are used in RNA processing in eukaryotes. So I also wanted to add that within eukaryotic cells, such as us humans, a primary transcript has to go through some extra processing in order to become a mature RNA. So initially, transcription produces a pre-mRNA molecule. So a molecule with exons and introns. So the pre-mRNA molecule has to be processed to become a mature RNA molecule, and it does this through caps that are added to the ends of RNA, and some pieces are carefully removed in a process called splicing. So here's your spliceosome. And the intron loops out as snRNPs, which stands for small nuclear ribonucleoprotein, which are produced from the snRNAs that we talked about earlier. And they bind to form the spliceosome right here. The intron is excised, and the exons are then spliced together. And the result is your mature mRNA. Oh, can't write. Where here is your excised intron loop, and your snRNPs can be released, and then this will release, your mature mRNA will release from the nucleus to go on to translation. So the last and final step is translation. This happens after transcription and is when an mRNA molecule is ready to direct protein synthesis. So during translation, the nucleotide sequence of an mRNA is translated into the amino acid sequence of polypeptides. So specifically, the nucleotides of the mRNA are read in triplets, groups of 3, called codons. One codon is a start codon that indicates where to start translation. And then another codon is a stop codon, which signals the end of a polypeptide. So this takes place inside of structures called ribosomes. These are molecular machines whose job it is to build polypeptides. And once a ribosome latches onto an mRNA, it finds the start codon, travels rapidly down the mRNA one codon at a time, and it will gradually build a chain of amino acids. So this happens in the cytoplasm. After the mRNA leaves the nucleus, and it happens in 3 steps called initiation, elongation, and termination. So here's your start codon, your stop, and everything that's in between are your RNA coding sequences. So within initiation, the components of the translational apparatus come together with an mRNA, a tRNA, and this carries the first amino acid. So here's your tRNA, your large ribosomal unit, your small unit, and then here is your first amino acid that the tRNA carries. So then In elongation, amino acids are brought to the mRNA by tRNAs, and they're added one by one to a growing polypeptide chain. So here's your growing polypeptide chain. Here are your tRNAs that are bringing the chains. And actually, a very common question is how ribosomes know which amino acids to add for each codon. And it turns out that it's not done by the ribosome itself. It's done by these tRNAs, or transfer RNAs. And these have 3 nucleotides sticking out at one end, as you can see right here, 1, 2, 3. And these recognize codons on the mRNA, and then they carry amino acids that specifically match those codons. So that's how they know which amino acids to add to the polypeptide based on the RNA, based on the mRNA codons. Smokey. Sorry, my cat decided that he wanted to be in the video. So we were talking about tRNAs. Usually they're just floating around in the cell, and only the tRNA that matches with the base pair codon is the one that binds and delivers the amino acid. So once a tRNA is snugly bound to its matching codon in the ribosome, its amino acid will be added at the end of the polypeptide chain, like right here, and then the tRNA will unbind and move on. And the last step is termination. So during termination, a stop codon in the mRNA is recognized by a protein release factor, and the translational apparatus comes apart, releasing a completed polypeptide. So here's your release factor. Here is your polypeptide chain that was growing. At your stop codon. And then once it releases, you have your completed polypeptide. So you can see where the translational apparatus comes together at the start codon, runs down the mRNA to add your your amino acids to the polypeptide chain, and then once it reaches your stop codon, it binds a release factor and releases the tRNAs and your completed polypeptide chain.